Hello everyone, myself Dr. Girish Adi, working as Assistant Professor in Department of Plant Pathology, School of Agriculture, SR University. Today I am here to discuss about integrated management of banana disease. Today I will discuss about some important diseases of banana and their integrated management using different IDM components like physical, mechanical, chemical, biological and other practices. Let's first start with the important banana disease and their causal organisms. First one is Panama disease or Panama wilt, which is caused by Fusarium oxysporum pharma species cubens. It is a very important significant fungal disease. Then yellow Sigatoka, which is caused by Mycospirilla musicola. There is another type of Sigatoka which is a black Sigatoka, which is caused by Mycospirilla fijiensis. Then another type of wilt, which is a bacterial wilt, it is also known as Moco wilt, which is caused by the bacterial pathogen Ralstonia solanisarum. Then banana bunchita, very important viral disease in banana caused by banana bunchita virus. Then anthracnose, it will cause the disease on the fruits or banana bunch, which is caused by Coltotrichum mysia. Then banana mosaic disease or banana mosaic virus, which is caused by cucumber mosaic virus. Then banana burrowing nematode, very important nematode disease in banana, which is Rhadophilus similis. Let's first start with the Sigatoka disease in banana. I already explained there are two types of Sigatoka. One is yellow Sigatoka and black Sigatoka. Mostly yellow Sigatoka, which is prevalent in India, and black Sigatoka is mostly not present in India. First coming to yellow Sigatoka, which is caused by Mycospirilla musicola, then black Sigatoka, which is caused by Mycospirilla fijiensis. Here the Pseudocercospora mysia is the imperfect stage of Mycospirilla musicola in yellow Sigatoka. Then Paracercospora fijiensis, which is asexual stage of Mycospirilla fijiensis. Let's start with the economic impact of Sigatoka disease in banana. This yellow Sigatoka, it is a worldwide in distribution, which is first observed in Java in 1902, and it is epidemic in 1913 in Sigatoka Valley in Fiji Island. Not reported from Canary Island, Egypt and Israel. In India, yellow Sigatoka is a serious threat to banana cultivation in states of Assam, Tamil Nadu, Karnataka, Andhra Pradesh, almost all the southern states of India. And then black Sigatoka, which is already not explained or not prevalent in India. Let's start with the symptomatology of Sigatoka disease in banana. Let's start with the first symptom. Early symptom appear on the third or fourth leaf from the top, that is mostly on young leaves. Then formation of small spindle-shaped spots on the foliage with a greyish center and yellowish halo, which is running parallel to the vines. Spots are mostly seen along the edge of the leaf with a defined margin and possess dark brown to black margin. Spot coalesce and whole leaf blade dries up. On the upper surface of the spots, we can see fructification of the fungus, which appear as a black specks. If the fruit is nearer to the maturity, then it is infected. In case of severe infection, the flesh ripens unevenly, that is uneven ripening of the banana fruit. And individual banana appears undersized and their flesh develops in a buff pinkish color and storage quality is very poor. Here you can see the picture representing the different types of symptoms of banana Sigatoka disease. The first symptom is development of small black color specks. Then in the picture see, you can clearly see spindle shaped spots with a grayish center surrounded by yellow zone or yellow halo, which is running parallel to the vines. Then all these parts, they will merge together. That will give like a scorching effect or burning effect or blighted appearance. In the last picture, you can see a complete burning appearance or blighting appearance of the infected leaf. Sometimes the infection may be confused with the potassium deficiency, because that will also cause marginal necrosis or marginal scorching effect. Coming to survival and spread of the Sigatoka pathogen. Survives and dry infected leaves in the field soil, and primary infection always takes place through ascospores, which is present in the infected plant debris. Secondary spread through wind-borne conidia and ascospore. Infection takes place through stomata on the lower surface of the leaves. You'll see the life cycle or disease cycle of Sigatoka disease of banana. In the picture, the first picture, you can see this is the infected plant. Is severely infected by Sigatoka fungus. Then formation of spots with the necrotic areas on the leaf. Then that necrotic area that will lead to formation of conidiospore and conidia, which is asexual structures of the fungi. We can clearly see conidia, conidia and conidiospore structure. These conidia of pseudocercospora, which are very long, thin, hyaline, slender, which are usually 5 to 7 celled, which are carried by the wind. Again, they will infect healthy leaves and they will cause infection. And also from the infected leaves, there is a formation of spermagonia or spermatia, which is nothing but a perithecia. Perithecia is a flask-shaped fruiting body. Inside it contains ascus and ascospore. Again, these ascospores, which are carried by the wind, again they will also cause infection on the healthy plant. Here you can see the germination of both ascospore and conidia. They will cause secondary infection. Then there is a lesion formation on the leaves on pattern of spotting on the leaf. Then severe spotting with a blighting appearance. So that how the fungus will continue the life cycle. So majorly you can have the role of spermatia, that is perithecia containing ascospore and conidia. Coming to integrated management of Sigatoka disease of banana, first management, removal and destruction of the affected leaves, followed by application of Bodos mixture 1% along with the linseed oil 2%. Then prevent water accumulation around the plant, go for periodical weeding. Spray cyanide, copper oxychloride at the rate of 0.3% suspended in mineral oil. Spray chlorothalonil or carbendazim or thiophanate methyl or mancozeb at the ratio of 0.1%. Very best effective fungicide is propiconazole at the rate of 1 ml per liter. While application of fungicide, we can go for spreading agent and wetting agents such as Tipal or Sandovit at the rate of 1 ml per liter. So these are some IDM practices which can use to control the Sigatoka disease in banana. Then start with the Panama wilt, which is also known as Fusarium wilt, which is caused by Fusarium oxysporum, former species cubens. First reported from Australia in 1876. The popular variety Grassmichael, mostly grown for export quality fruits, were the most susceptible and are replaced with the Cavendish banana, which were resistant to Panama wilt. Severe and Rasatali variety, that is Amrutapani, Grassmichael, Monthan, Karpuravalli. Let's start with the most diagnostic symptom of Panama wilt. The young leaves may not dry up but they remain erect. They also get affected under severe cases. There is a odd leaf alone remains upright and the remaining leaves they will turn to yellow color, that is sclerosis, and they will hang down from the main axis. The symptom initially seen on the older plants in the mat and older leaves. Leaves show progressive yellowing, break at the petiole and hang down from the pseudostem is the main diagnostic symptom of Panama wilt. These are the another diagnostic symptom. At the base of the plant, we can see there is a V-shaped splitting or crack development on the base of the stem. And when we cut open the infected plant, we can see brown to black color discoloration of the pseudostem near the periphery region, which is more concentrated on the periphery region. So again, these are the symptoms: light yellow to dark brown discoloration of the pseudostem. Usually discoloration appears first in the outer or oldest leaf sheath and extended up to pseudostem. Varying degree of longitudinal splitting along the pseudostem are often observed, and the infected plant will emit a characteristic odor of rotten fish. So these are the important diagnostic symptom of Panama wilt or Panama disease in banana. Coming to favorable condition, soil conditions like texture, structure, fertility level, temperature, drainage, cultural operation greatly influence the disease incidence and spreading in the field condition. Survival and growth of the fungus are generally greater in acidic or light textured soil than in clay or alkaline soil with a high calcium content. Saturated, poorly drained soils have greater incidence when compared to dry soil. Insect injuries, wounds, especially the furrowing nematode, Rhadophilus similis burrowing nematode will act as a predisposing factor. Here we can see the life cycle of Fusarium oxysporum, former species cubens. Mainly the pathogen will survive in the infected planting material or banana sucker. This banana sucker infected by Panama wilt, which is act as a major carrier of infection. When the farmers use infected suckers for planting, there is a development of the disease. There is a movement of pathogen from the root zone to top portion of the leaf through xylem tissue, then formation of the resting spore and complete drying and wilting of the plant, then complete death of the plant will occur and the fungus will return to resting spore. That is, mainly this fusarium will produce microconidia, macroconidia and chlamydospores. These chlamydospores are essential double-walled retic-walled structure which are able to survive in the soil for 3 to 4 years. Then these micro and macroconidia, they will spread the disease. They will act as a secondary spreading agent. Coming to management of Panama disease or Panama wilt, use the disease-free suckers for planting. Best go for tissue culture banana. Avoid yield-drained soils, perfectly slightly alkaline soil with a pH of 7 to 7.5 for cultivation. Flood following for a duration of 2 years, that is 6 months to 2 years is recommended. Along with the crop rotation, you can go for banana after rice. Application of lime at the rate of 1 to 2 kg per pit at the infected pits after chopping of the plant parts. Dipping suckers in carbendazim solution 1.1% rate before planting. Soil drenching with a 0.1% carbendazim or Vepam. Growing resistant varieties like Cavendish varieties, Basarai, Puvan, Mungal, dwarf varieties are generally resistant to Panama wilt. So these are some IDM practices recommended for the control of Panama disease or Panama wilting banana. Let's start with another important fungal disease. Mainly this disease occurs on the banana fingers or banana fruit, which is known as Siger end rot of banana. The Siger end rot, which is caused by Verticillium theobrome. It is a very important fungal disease on fruits. A black necrosis spread from the perianth into tip of the immature fingers. Mainly the tip of the immature fingers get infected by Verticillium theobrome. The pulp of the affected fruit undergoes a dry rot. The dry rot can spread up to 2 cm from the tip of the banana fingers. The necrotic tissue becomes covered with the fungal mycelia or resembles the grayish ash of the Siger end. So that's why the disease name itself we call it as Siger end rot. If emerging fingers are affected, the entire finger sometimes get rotten. So this is the complete rotting of the tip or tip end of banana fingers caused by Verticillium theobrome. Let's we know about disease development. The disease incidence is high during the summer season. The fungus is commonly seen in the leaf trash or the flower, which will survive in the infected plant debris, and conidia disseminated in the air current and infect the flower parts. Then eventually the banana fingers are get infected by Verticillium theobrome. And let's see about important management practice for the Siger end rotting banana. Field sanitation is very important. Removal of infected plant pots. Then plantation should have enough aeration. Go for optimum spacing is very important. Usually banana is cultivated with a spacing of 6 by 6 feet, 7 by 7, 8 by 8, 9 by 9 they are following. 6 by 6 is a very high density planting. Better to go for 6 by 8 or 6 by 7 spacing. After emergence, the bunches are covered with a polythene bag. Better avoid the infection, and packing and ripening room need to be kept very clean because it will also act as a post-harvest pathogen. Spray of mancozeb at the rate of 75% wettable powder at the rate of 1.5 to 2 kg per hectare. This mancozeb is a contact fungicide which can act on the Verticillium theobrome. So this is regarding symptoms, etiology and management of Siger end rot of banana. So this complete the important fungal disease in banana. Let's start with the bacterial infections in banana. First we'll go for Moko disease or bacterial wilt in banana, which is caused by Ralstonia solanisarum. So this disease first reported in Guyana in 1814, Moko plantain region. In 1968, this reported first from West Bengal in India in 1968. Let's start with the important symptoms of Moko wilt of banana caused by Ralstonia solanisarum. The youngest 3 to 4 leaves turn pale green or yellow and collapse near the junction of lamina and petiole. And characteristic discoloration of vascular strands, wilting and blackening of the suckers will occur. Vascular discoloration, the pale yellow to dark brown discoloration, which is mostly concentrated on the center of the pseudostem, wherein Panama wilt, which is mostly concentrated on the periphery region, becoming less apparent in the periphery. Grayish brown bacterial ooze formations may be seen on the pseudostem when we cut the affected plant transversely. A film brown dry rot found within the fruit of infected plant, which is the most noticeable symptom, dry rot of infected banana fingers. Death of the whole plant may occur under severe condition. This Moco wilt is caused by race 2 of Ralstonia solanacearum. These are the symptoms of Moco wilt of banana. In the first picture, we can see a brown to black color discoloration of the pseudostem near the center region. And when we cut open the infected plant, we can see bacterial ooze formation. So these are the banana fingers. We can see the dry black color rotting or discoloration of banana fingers. So these are collective symptom of Moco wilt of banana. Coming to disease cycle, the primary infection always occurs through disease suckers or disease plants used for planting. Secondary infection may occur through irrigation water, implements, and by insects also. They will transmit the pathogen from infected to healthy plant. Coming to management of Moco wilt of banana, grow resistant varieties like Puvan, Montan, adopt strict quarantine and phytosanitary measure. This will avoid the movement of banana sucker from infected area to healthy area. We can prevent 50% of infection by avoiding these infected planting material. Use of healthy planting material, exposure of soil to the sunlight during dry hot weather, that is go for deep summer plowing, soil solarization activities during in the month of May, June. Eradicate the infected plant and suckers, killing in situ by application of herbicides, which will avoid the infection. Disinfection of the tools with the formaldehyde, like when we are removing the banana suckers or any agricultural activities, go for disinfection of the tools by using formaldehyde. Then we can also go for crop rotation, 3 years of crop rotation with the sugarcane or rice, and providing good drainage practice, which will help to avoid the disease infection. Allow fallow period or flood following during off season. Fumigation of infected site with the methyl bromide or chloropicrin. Spray systemic insecticide to prevent the transmission of disease to healthy plant. And we can also go for biocontrol application of Pseudomonas fluorescens, which may applied along with the planting. You can mix with the farmyard manure or vermicompost along with the oil cakes like neem cake, pongema cake, or any other oil cakes, which is having antimicrobial properties. So this complete the Moco wilt or Moco disease in banana caused by Ralstonia solanacearum. Let's start with another important disease, Erwinia rhizome rot. So this Erwinia rhizome rot, it is also known as tip over disease in banana or bacterial rot, which is caused by Erwinia keratovera subspecies keratovera or Erwinia chrysanthemi or Pectobacterium keratoverum. So here the affected plant, they will show discoloration and soft rotting of rhizomes and suckers. The affected plants have scanty root system with a dark brown lesion and necrotic tip. In many cases, the pseudostem tip over breaking across the rotor stem, particularly at the ground level. That's why this disease is known as tip over disease. So coming to the symptoms of Erwinia rhizome rot or tip over disease in banana, here the mature plants show above ground symptoms until the disease is well advanced. Such plant may produce small size of fruits which fail to emerge from the shoot tip. Infected plants can be pushed over easily. We can pull the infected plant easily because which are very susceptible to wind damage and also which is having very scanty root system. In severely infected soil, newly planted rhizomes may rot and fail to sprout. When the young plants are infected, dark brown necrosis appears on the lamina of the older leaves and later plant becomes stunted with a yellow coloration. As the rhizome become infected, pockets of dark water-soaked areas may develop on the infected rhizome or infected sucker. Infection may be result in production of cavities, which is most resemble like the root borer tunnels caused by the root borer. These are the pictures representing Erwinia rhizome rot. Here the yellowing and wilting of the older leaves, they will hang down from the main stem. Then brown color discoloration and bacterial ooze formation on the banana infected fingers, and also like complete rotting of the banana suckers or banana rhizome. Then tip over will occur. That's why it is known as tip over disease. Coming to the primary source of infection, it is always occurs through banana suckers used for planting, or infected soil will act as a primary source of infection. Secondary infection, it will also spread through irrigation water, implements, and through insects. Coming to favorable condition, prevalence of high temperature during initial stage of crop growth and growing varieties like Vanamakeli, Basarai and Tilla Chakkarakeli which are resistant to Erwinia rhizome rot. Coming to management of Erwinia rhizome rot, avoid planting susceptible varieties during periods of high rainfall or in waterlogged soils in a problematic area. Maintain proper soil aeration by intercultivation operation. Ensure that only healthy suckers are planted. Restrict the ratoon 1 or 2 based on the disease incidence. Crop rotation with rice or sugarcane. Remove and destroy infected plant along with the roots. Dip the suckers in copper oxychloride 5 gram per liter plus monocrotopus solution. Drench the infected suckers with a bleaching powder at the rate of 25 gram per liter of solution. Let's start with viral disease in banana. There are three important viral disease. First, let's start with the bunchy top disease in banana. This bunchy top or curly top or cabbage top or strangle disease which is caused by banana virus 1 or Musa virus. First reported from Fiji in 1889 in Cavendish varieties. Around 1940 introduced to India from Sri Lanka through cyclone. Banana bunchy top virus is a single-stranded DNA virus with a single isometric particle. So these are the characteristic diagnostic symptom of banana bunchy top disease. Coming to the symptom, dark brown broken bands of greenish tissue on the vines and leaves and petiole region. Plants are extremely stunted, leaves are reduced in size, marginal chlorosis and curling of the top. Leaves upright and become brittle. Many leaves are crowded at the top and resemble a bunchy top appearance. Bunches are very small in size. If infected early, no bunch formation will occur. So in this picture, you can say formation of marginal chlorosis and curling of the leaves, completely bunchy appearance, and green jay books and moss coat appearance, and mosaic appearance of the plant. And this is severely stunted plant with a curly top. And sometimes the infected plant there is no bunch formation will occurs. Coming to etiology of banana bunchy top virus, the primary infection takes place through virus particle present in the disease suckers. Secondary infection, the virus is transmitted by banana black apid Pentalonia nigronervosa. Colocasia esculenta serve as a latent reservoir of host. This pathogen virus contains single-stranded DNA virus with an isometric particle as a genome. Coming to management of bunchy top disease in banana, adaptation of strict quarantine measure, eradication of all infected sucker by spraying with the kerosene or by injecting herbicide like 2,4-D. Use of only certified banana suckers for planting. The new crop should be regularly inspected and disease plant destroyed as soon as noticed. Vector control with a systemic insecticide like paspomidone, methyltimidone or any other systemic insecticide we can recommend to control the banana black apid. Coming to another important viral disease which is banana bract mosaic disease. So this banana bract mosaic virus which is first reported in Philippines in 1979, recorded in Sri Lanka, India, Vietnam and Philippines. In India, this is first reported from Kerala. Initially it is known as Kokan disease in banana. Very rapidly spreading in all the banana growing states in India in absence of, there is no clear cut regulation for the movement of banana sucker from one state to another state. So strict quarantine measures are actually needed to control the viral diseases in banana. Let's start with the symptom of banana bract mosaic. The symptom expression is clearly seen in montan and plantain group of banana. The disease is characterized by the development of black streak on the petiole, yellow or pinkish coloration on the pseudostem, mosaic like purple and spindle shaped streaks on the bract. In severe cases, pink streaks continue on the lower surface of the midrib. Such sucker, they exhibit unusual reddish brown streaks at the emergence and separation of the leaf sheath from the central axis. And also infected plant, the leaves cluster at the crown region and the leaf sheath separates from the central axis. And the clustering of the leaves at the crown region with a traveler's palm appearance is the most characteristic symptom of bract mosaic disease in banana. Then off-filled fingers like ants which are the characteristic symptom of banana bract mosaic. Coming to the diagnostic symptom, you can see streak on the bunch stalk and streaks or like pinkish color streak development on the inflorescence and like the pinkish color streak development on the pseudostem and elongated and brittle peduncle with a traveler's palm appearance. So coming to etiology, banana black mosaic virus, the primary infection takes place through viral particle, vertically spreads through disease sucker used for planting, and this genus belongs to partyvirus in the family of Partiviridae. Coming to secondary infection, virus particles spreads through transmitted by banana aphids, Apis gossypi, Apis maedis and Rapulus maedis in a non-persistent manner. And the long fleshless rods which is having single stranded RNA is a genome of banana bract mosaic virus. So coming to management of banana bract mosaic virus, early detection and roguing out of infected plant. In all the viral disease, early detection and roguing is important strategy to avoid the viral infection. Use of disease-free suckers for planting, we can avoid infected plants. Better to go for tissue culture banana because these tissue culture banana which are lack of any kind of viral and fungal infection, and also we can go for dry heat treatment of suckers at 40 degrees Celsius for 1 day. Use of seedlings is to tissue culture technology. Raising a barrier crop like sunhemp in 3 to 4 rows in the field boundaries that will check the movement of vector from one field to another field, and we can control the vector with application of methyl dimethan, dimethoate or any other systemic insecticide to control the aphids vectors which will transmit the banana bract mosaic. So these are the symptoms, etiology and management of banana bract mosaic virus. Then coming to banana mosaic or infectious sclerosis. Banana mosaic or infectious sclerosis, another important viral disease which is caused by cucumber mosaic virus, which is having the genome single-stranded RNA virus. So these are the infected plant which are showing the symptom of mosaic or infectious sclerosis along with the heart rot, that is rotting of heart leaf. Coming to the symptom, typical mosaic like discontinuous linear streaking bands extending from mid margin to the midrib like mosaic pattern, and leaf size is extremely reduced and leaves are malformed, rolling of the leaf margin, twisting and bunching of the leaves at the crown and a rigid erectness in newly emerged leaves. Chlorosis of newly formed leaves like infectious sclerosis, rotting of heart leaf and leaf sheath in severe cases which is known as heart rot symptom, which progress into pseudostem and plant will die within a short span. Diseased plant do not reach maturity, there is no bunch formation, there is no inflorescence formation. Coming to banana mosaic or infectious sclerosis, let's see the etiology of this virus. The causal agent of this disease is cucumber mosaic virus (CMV). The virus is isometric, a linear positive sense single-stranded RNA. The primary transmission through use of infected daughter suckers from the diseased plant, and the secondary spread is through Apis gossypi and Apis maedis, which is a melon aphid. Then coming to management of banana mosaic or infectious sclerosis, the banana garden should be kept free from any kind of weeds. Use virus indexed certified tissue culture banana for planting, which will avoid all kind of viral and bacterial infection. Infected suckers should not be used for planting. While you are using any kind of suckers for planting, avoid the selection of infected suckers. And growing pumpkin, cucumber and other cucurbits between the rows of banana crop should be avoided because it is a cucumber mosaic virus which will also infect this pumpkin, cucumber and other cucurbit varieties. Early detection by regular inspection of planting and eradication of diseased plant from the field, which is a very best strategy to avoid the infection. Use of high input crop management of system comprising 10 kg of farmed manure at the time of planting and subsequently at the interval of 3 months, or else we can go for 1 kg of neem cake, 2 gram of 200 gram of nitrogen, 40 gram of phosphorus and 200 gram of potassium per plant for weeding at 2 months interval up to 8 months stage. These are some agronomic practices which will avoid the infection, which will increase the resistance in the banana plant. And application of systemic insecticide like dimethoate 30 EC at the rate of 1 ml per liter and methyl dimethion 25 EC at the rate of 2 ml per liter, we can control the vector movement. These are the some of the important fungal, bacterial, and viral disease in banana. Like in case of fungal disease, Panama wilt and Sigatoka disease are highly important disease. Coming to bacteria, Moco wilt and Tipor disease caused by Erwinia, which are highly important bacterial disease. Coming to viral disease, like infectious sclerosis, they are caused by cucumber mosaic virus and banana bunchy top, and other viral diseases are very much important. So these are the some objective questions related to Panama, diseases in banana and their management. Let's start with the first question. Panama disease in banana, it is caused by options are for bacteria, virus, fungus, and nematode. The correct option is fungus. The causal organism of Panama disease, which is nothing but Fusarium oxysporum, Pharma species cubans. A key symptom of Panama disease. There are many symptoms like leaf spot, yellowing and wilting, fruit rot, stem canker. The best symptom is a suitable answer is yellowing and wilting of the leaves. And then Sigatoka leaf spot is primarily caused by bacteria, fungus, virus. It is a very important fungal disease. Black Sigatoka is caused by, already Mycospirilla fusionensis. Which practice helps to manage the Sigatoka disease? Like removal of infected leaves, which is a best answer. Bunchy top disease in banana transmitted by, already banana black apid, Pantalonia nigronervosa. The causal agent of banana bunchy top is a fungal, bacteria, virus, or nematode. The suitable answer is virus. A common symptom of bunchy top disease in banana, bunchy appearance or curly top or cabbage top or bunchy appearance of the infected leaves. Control of bunchy top in disease includes like removal of infected plant. And banana bacterial wilt, it is caused by, Ralstonia solanisarum. The major symptom of banana bacterial wilt is like oozing of bacterial slime, oozing of bacterial liquid from the infected cut end of the banana pseudostem. And anthracnose in banana, which mainly affects root, leaves, fruits, and flower. Correct answer is fruits. The causal organism of anthracnose is, Coltotrichum mysia, which will cause anthracnose disease in banana fruit. Proper post-harvest management of anthracnose includes like hot water treatment we recommend to control the post-harvest management of anthracnose. Banana mosaic disease is caused by, it is an important viral disease, which is an effective strategy for the viral disease in banana. Use of disease-free planting material like tissue culture banana. Leaf speckle disease in banana, which is caused by Philostricta miserium. So these are the some important objective questions related to different diseases on banana and their integrated management. Thank you.